Okay, so here we are having the characteristics of the enzyme. What does it mean? It means that we will be discussing about some character of the enzyme. So as you guys all know, in introduction I told you that most of the enzyme are protein, but some of them are non-protein, that is ribozyme. But most of the enzyme are proteinaceous in nature, but some of them are non-protein too. Okay, but some enzyme are purely protein too, purely protein in nature. What is purely protein? That are the simple protein, or you can say that simple enzyme. Okay, but some have little part of the protein with non-protein with protein part. Means that some have non-protein part as well as protein part, like protein part and non-protein part. These type of enzyme are called conjugated enzyme. What does it mean? They are also called they also act as simple enzyme. They are called holozyme. What is holozyme? Holozyme is an active enzyme. Okay, they're composed of protein. Okay, like apoenzyme and cofactor. Will be discussing this too. So conjugated enzyme are consisting of two parts, that is the protein part, as you guys all know, and as well as non-protein part. What is protein part? In protein part there is apoenzyme. What is apoenzyme? This is the protein part enzyme which is bind with, or you can say that this is inactive. This is inactive. Okay, and they are bind with cofactor prostatic, it will become active too. Okay, so this is protein. Non-protein, that is prosthetic group. What is prosthetic group? Okay, and what does it mean? It is inactive. Inactive means like that we are having pepsinogen. This is inactive enzyme, and after the HCl will bind with it with them, and they will like you can say that it will HCl will, you can say that through the HCl the pepsinogen will convert into the pepsin. So this is the apo enzyme that is inactive, for like for example pepsinogen. Okay, and here you can see that holoenzyme means that they are an active enzyme. Okay, so if you're talking about what's the example of the purely protein, so the purely protein is the amylase. This is purely protein. And if you're talking about holozyme, that one is non-protein part and one is protein part, so we can take the example of amylopectin too. Okay, so what's about prosthetic group? So what is prosthetic group? Prosthetic group consists of organic, non-protein and inorganic. No, they're having inorganic. Okay, so this is based, what is the over on in organic, that is coenzyme. Here the coenzyme is there. What is coenzyme? That helps the enzyme or to continue the activity. For example, NAD, nicotinamide diphosphate. And in inorganic we are having cofactor. What is cofactor? Cofactor means the activator. Like I have told you in the previous video, there will be salt that dissolve. Okay, for example magnesium, zinc we are having. Okay, so we are having two type of enzyme. One is the endozyme. Okay, so what is endozyme? Endozyme means that endozyme, that cofactor is the protein part. Okay, and this is non-protein. Okay, So here you can see that we are having the two type of enzyme, that is endozyme. Endozyme, what is endozyme? That enzyme that act within the cell or inside the cell, that is called a endozyme. For example, we are having a adenylate cyclase. This is a endoenzyme, which there is an enzyme that work in any hormone or substrate that become active. Okay, what is substrate? That for example we are eating, that is substrate, for example. And we are having the enzyme that will be you can say that large. Okay, so we are having, for example, we are having food. This is our food. We are, for example, we are taking them as a substrate. This will be having a small size, but enzyme will be having a more size, we can say that. But in the next video I'll be telling you that too. Okay. So you can say that we are having a, you can say that we are having the uh, adenylate silase that help in the hormone to become active inside the cell. Okay, so we are having you can say that, for example, we are having a structure that is the like this is, you can say, that active site through which the enzyme will bind with that. Okay, for example, we are having one protein here, one carbohydrate there, and one, you can say, another molecule. So it will getting inside easily. Okay, like this. So this one is this protein. Okay, so that is the binding protein, and that is the active site. Okay, active site is that will bind with the molecule that is binding with the active site. Okay, so we are having the binding site, and one is the catalytic site, which means that catalytic is constituted with each other. Okay, so okay, so we are having, one is endozyme, the second we are having that is the, you can say, that is exozyme. The exozyme will be opposite, that it will be act outside the cell. Okay, for example, all digestive enzyme, they act outside the digestive system. Okay, so the size of enzyme is greater and the size of substrate is lesser. Okay, here we discuss this. Okay, so enzyme are specific in nature, they are having different function. Every enzyme are having different activities, they are having different pH, but the most point of the enzyme is that they work, every enzyme work on different pH. Okay, so some enzyme are alkaline, some are acidic. For example, acidic enzyme are present in stomach, and alkaline enzyme are present in small intestine, large intestine. So the stomach enzyme will not work in the large intestine or small intestine. Why? Because they are having acidic pH, and in small intestine and large intestine, the enzyme are work as alkaline, so they are having the pH of the enzyme. Every enzyme is having different pH and different, they work on different chemical reaction. So there are many enzymes in our body. Okay, so one, we are having a terminology that is inhibitor. What is inhibitor? Inhibitor means that the that stop the activity of enzyme. That is you can say that as a inhibitor. So today we are done with the characteristics of the enzyme.